Good evening. My name is Uma Mageshwari. Here we can discuss about the topic of transgenic plants and animals, which controls under the which comes under the topic of biology of its for its industrial application and in the subject of biology for engineers. Here, in this case, like how the introduction to transgenic organisms, how these transgenic organisms are generally engineering into incorporated and then foreign DNA is from the another specific using a recombined DNA technology. Here, this is the thing we are just going to discuss over here. This genetic modification introduces a new desired triad such as diseases resistance in plants like cotton or the production of human proteins in the milk of transgenic cows. Especially we know very well that cows, that is the transgenic cows, is the main thing. Like here the application includes here creating the animal models of disease research, improving the crop yields and then developing the biological products and then testing the vaccine safety. What are all the methods of creating the transgenic plants? Here we'll just discuss this. Like the main approaches is transgenic plants are created using genetic engineering methods such as agrobacterium mediated transformation and then gene gun method. What is that gene gun method into plant cell? How it has been processed? The general process involves designing a gene conversation that has been concern that has been introducing into the plant cell via a vector like a agrobacterium TI and then or the gene gun selecting for the transformed cells using marker genes and then regenerating the whole new plants from these cells that express the new trait. Key methods for gene transfer is it is a mechanism. The methods of creating a transgenic is it is a mechanism and it is also a process. The method used the soil bacterium aquaterium is which naturally transfers its own DNA, and the process is that like the scientist inserted the desired gene into an aquabacterial TI, that is a tumor introducing plasmid. The modified bacterium then also that has been infected the plants, delivering the foreign gene into the plant's chromosome. The applications are this has been widely used under the effective method for many plant species and including crop like wheat, rice, etc. How much the process, that is the application of transgenic plants, before this topic we'll just see what is the process for this one. The small particles of gold or tungsten coated with the foreign DNA are shot into the plant tissues and then cells at high speed using the device called a gene gun. The application is this method is versatile and can be used for plant and that are difficult to transfer the wheat, and then this thing that the electropolarization uses the electric current to create temporary pores in the plant cell membrane, and it is been allowing the DNA to enter. So the methods, everything we have seen now so far. The application is like the medical research says that the models of human diseases, like it is a knockout mice. The transgenic plants are genetically engineering to provide valuable traits like and products with the major applications including enhanced resistance to pests diseases and then improved tolerance to epitotic and then stresses like drought and then salinity and then elevated nutritional content. And the key applications are stress resistance, biotic stress resistance, that is in engineering plants to resist infections from viruses, that is the fungus, bacteria, and then infectious. And then the future of this transgenic technology are like the benefits are we have seen so far, and the future is like nothing but like the future genetic technology is the potential development, more nutrition enhanced crops to combat the global malnutrition. Here the types of ethical concerns are like these are all the concerns we just taken into account. Like it is a honesty and integrity, upholding the resistance delightfulness practices, and the challenges and the benefits is nothing but it is a quality of the whole food and the specific nutrition composition. The transgenic technology could provide a means of transferring or increasing the nutritionally beneficial trials. So these are all the methods of creating transgenic animals like microinjection, viral vectors, and then CRPIS. And the future prospects are the improved crop reliances to climate change, and then advanced medical therapies, sustainable agriculture, and then environment, and then challenges are the public acceptances, etc. Here, these are all the what is the genetic. The previous slide shows the um, activity of the thing, and this is the previous slide. The previous slide shows the activity, and the assessment questions are four over questions are here, and the remaining four questions are over here. What are the major applications? So, I mean, we have seen in this PPT. So, thank you. You can go through these questions for a better experience. Thank you. We see the next topic in the next video. Thank you.